Thyroid hormone secretes two major metabolic thyroid gland secretes two major metabolic hormones. One is the thyroxine, which is known as T4, and other is triiodothyronine, which is T3. Both of these hormones have profound effect in increasing the metabolic rate of the body. If there is complete lack of thyroid hormone secretion, the BMR falls to almost 40 to 50% below normal. And if there is extreme excess of the thyroid secretion, the BMR can increase up to 60 to 100% above normal. The thyroid gland also secretes a very important hormone for calcium metabolism, which is known as calcitonin. Its secretion is controlled by mainly a hormone secreted by anterior pituitary gland, which is thyroid stimulating hormone. We'll study it in detail in the regulation of thyroid hormone secretion. Now let's have a look at the physiologic anatomy or histology of the thyroid gland. If you look at this diagram of the thyroid, histologic diagram of the thyroid gland, you can see that it consists of a large number of closed follicles. These are the large follicles, about 100 to 300 micrometers in diameter. They are filled with a secretory substance which is known as colloid, and these follicles are lined with cuboidal epithelial cells. These cells are very important because they secrete their secretions into the interior of the follicles, and the major part of this secretion is a glycoprotein which is known as thyroglobulin. This secretion is known as colloid, and it contains thyroglobulin, which is very important for the synthesis of thyroid hormones. This is the histologic view of the thyroid gland. It consists of large numbers of closed follicles, 100 to 300 micrometers in diameter. These follicles are filled with secretory substance called colloid. They are lined with cuboidal epithelial cells that secrete thyroid hormones into the interior of the follicles. The major constituent of colloid is the large glycoprotein thyroglobulin, which contains the thyroid hormones. Thyroid gland also houses important endocrine cells nestled in the spaces between thyroid follicles. These cells are known as parafollicular cells or C cells, which secrete the hormone calcitonin. If you look at this view, this is a large follicle. In the middle, in the center, it is filled with colloid. The colloid contains a major constituent which is thyroglobulin. It is lined by a cuboidal epithelial cells. These cells are important in secreting thyroglobulin into the cells, and all the other functions we'll study it in detail later on. Here are also the parafollicular cells we cannot see in this. The parafollicular cells are present like this in the spaces between these follicles. If there is another follicle, a big follicle here, the spaces between the follicles are filled with the cells which are known as the C cells or the parafollicular cells, and these cells secrete calcitonin. If we quickly have a look at this table, it listed the different parts of the thyroid gland along with their function or description. The thyroid is composed of spherical follicles that selectively absorb iodine as iodide ions from the blood for the production of thyroid hormones. Inside the follicles, colloids rich in a protein called thyroid globulin serve as a reservoir of materials for thyroid hormone production and to a lesser extent act as a reservoir for the hormones themselves. The cells of the thyroid follicles are the cuboidal cells, epithelial cells. They are surrounded by a single layer of thyroid epithelial cells which secrete T3 and T4. Parafollicular cells or C cells are the scattered among follicular cells in spaces between the spherical follicles. They are another type of the thyroid cells and they secrete calcitonin. The thyroid gland has a blood flow about 5 times the weight of the gland each time. Unlike other endocrine glands which secrete their hormones once they are produced, thyroid gland stores considerable amount of thyroid hormones in the colloid until they are needed by the body. They store a large amount of thyroid hormones. This is a special feature of the thyroid gland. Now, the synthesis, storage, and secretion of thyroid hormones. The raw material for the synthesis of thyroid hormones are the tyrosine, which is an amino acid, and iodine. These two are important raw materials for the synthesis of thyroid hormone. Stages of thyroid hormone synthesis. The first stage is the synthesis and release of thyroglobulin. The thyroid cells are typical protein secreting glandular cells, and the endoplasmic reticulum and Golgi apparatus of these cells synthesize and secrete thyroglobulin into the follicles. It's a large glycoprotein, and it is made up of about 70 tyrosine amino acids. As I have already told you that tyrosine is the raw material for the thyroid hormone synthesis. So the tyrosine is present in the thyroglobulin. About 70 tyrosine amino acids are present in a single molecule of thyroglobulin, and they are the major substrate which combine with iodine to form the thyroid hormones. After the synthesis of thyroglobulin by these cells, by the thyroid follicular cell, the cuboidal epithelial cells, it is secreted into the follicle, into the center of the follicle, central cavity of the follicle, which is filled with colloid, and by the process of exocytosis. This is the first step of thyroid hormone synthesis, that is synthesis of thyroglobulin. It is synthesized by endoplasmic reticulum, the protein synthesizing machinery which is present in each cell. Similarly, like other all other proteins, it is also synthesized by transcription and translation processes, and this thyroglobulin is then released by exocytosis into the colloid. The next thing is the iodide pump, that is the transport of iodide ions from ECF or from blood into the thyroid glandular cells and then into the follicles. This is known as iodide pump. The iodide pump, the basal membrane of the thyroid cell has the specific ability to pump the iodide actively to interior of the cell. This pumping is achieved by the action of a thyroid pump, the iodide pump, that is sodium iodide symporter. This pump co-transports one iodide ion along with two sodium ions across the basolateral membrane into the cell. And the energy for transporting iodide against a concentration gradient comes from the sodium potassium ATPase pump, which pumps sodium out of the cell and thereby establishing a low intracellular sodium ion concentration and gradient for facilitated diffusion of sodium ions into the cell. This process of concentrating the iodide in the cell is known as iodide trapping. After trapping of iodide into the cells, this iodide or iodide ions are transported into the follicles, into the center of the follicle by another channel, which is a chloride iodide counter transport channel, and this protein is known as pendrin. It transports chloride into the cells and similarly at the same time iodide ions into the follicle, into the colloid. In a normal gland, the iodide pump concentrates iodide to about 30 times its concentration in the blood, and this is inhibited by thiocyanate and perchlorate anions. Now this is the second step of the thyroid hormone synthesis. There is a symporter which is known as sodium sodium iodide symporter. It is present on the basolateral membrane of the thyroid follicular cells, and it transports iodide ions and sodium ions both in the same direction from the ECF into the cell. From the into the cell, this is known as iodide trapping. From the cell, there is another channel or another protein which is known as pendrin, and it is located on the luminal surface of the thyroid follicular cell. It transports chloride ions to the inner of the cell, and at the same time iodide ions from the cell into the follicle, into the center of the follicle, the follicular cavity, or we can say into the colloid. This one. When the thyroid gland becomes maximally active, this concentration ratio can rise to as high as 250 times. The rate of iodide trapping by the thyroid is influenced by several factors, the most important being the concentration of thyroid stimulating hormone, which is being secreted by the anterior pituitary gland. If the anterior pituitary gland is removed, the synthesis of thyroid hormone decreases to a very low level, and because of the decreased activity of thyroid, iodide pumps in the thyroid cells.